Good morning and welcome to Biology 102. Today we're talking about macroevolution. Macroevolution is defined as evolution at or above the species level. Macroevolution allows us to talk about the origin of new species and what really creates the great diversity of species on planet Earth. You can see all these beetle species. So it answers questions like why and how did all of those beetle species evolve on the surface of the Earth. In order to really understand macroevolution, that is evolution at or above the species level, you have to understand what a species is. And a species actually has three different components to it. A species are population whose members can interbreed. They can do this in nature without the help of scientists, and they produce fertile offspring. So those are the three qualifiers to be of one species. Our species definition is based on reproductive capabilities and compatibilities versus physical appearance. You can see over here these birds, these western and eastern meadowlarks, to most people's eyes look the same, right? But they are two separate species. So we base this definition upon whether you can interbreed with something in nature and produce fertile offspring. Now, what keeps species like these meadowlarks from interbreeding accidentally, right? So what kinds of things do that? The kinds of things that keep species that are closely related from interbreeding, those things are called reproductive barriers. And these are barriers that isolate one population from another and keep them from breeding on accident. This keeps things like these hybrids from occurring in nature. And so usually these hybrids are typically forced by humans. So what kinds of reproductive barriers and what are some examples of those reproductive barriers that we see in nature? So there are two groups of reproductive barriers, and one is called pre-zygotic barriers and one is called post-zygotic barriers. You have to know what a zygote is, and a zygote is the first cell formed from the union of a sperm and an egg. So when we have the union of the sperm and the egg, we form that first cell. If a barrier prevents mating or fertilization, prevents the formation of a zygote, then it is a pre-zygotic barrier. If the zygote forms and then development doesn't work out or the species kind of falls apart after, then that is called a post-zygotic barrier. I think it's easiest if I give you some examples of each of these so that you can see how this works. So pre-zygotic barriers are going to prevent mating and fertilization from happening in the first place. So we're not going to form that zygote. This can be temporal barriers like these skunk species that breed at two different times of the year, one in late winter, one in sort of spring, early summer. And if you're breeding at two different seasons or two different times, then this isolates you or keeps you from accidentally breeding with the wrong species. Habitat isolation are like these garter snakes. Some live in the water, some live primarily on land. They don't really see each other because they're in these two different habitat types, so this keeps them separated. Behavioral isolation, like the blue-footed booby does a little dance, can separate species so that if you're closely related, you're not attracted to certain features and certain behaviors. Mechanical isolation is where two species don't fit together. A good example of this is if you crack open some snail species that look almost identical on the outside, you will find that some of the snails run clockwise and some counterclockwise. When they get together, then they cannot sort of fit because of this internal shell rotation, and they cannot fit, and so the delivery of the sperm and egg doesn't work out. And lastly, gametic isolation is where the sperm and the egg, the chemicals on the outside surface, either don't attract the two together or the two cannot fuse chemically, and so you don't get the zygote forming. Post-zygotic barriers are those where you allow the zygote to form, but then something else falls apart soon after that. So a post-zygotic barrier might be like this salamander species. This is called hybrid inviability, where you form the zygote and it grows into a juvenile salamander. These are two related species that will mate and form this kind of frail offspring, but the offspring doesn't live to the age of reproduction. So it always dies before it reaches that age of reproduction. So we say that's hybrid inviability. There's also hybrid sterility. Horses and donkeys can be forced to mate and create mules. You might have seen this on farms or heard about this, but the chromosome numbers are quite different. So horses and donkeys have 2 different chromosome numbers, and then mules get an in-between number. This causes sterility or problems with them mating with other mules. And lastly, hybrid breakdown is like in these particular grains. This is showing rice here, but what happens is sometimes we'll get the first generation will be kind of frail, but it will make it to reproduction, but the second generation falls apart. So this is called hybrid breakdown, where you make it through kind of a stunted first generation or problematic first generation. You get to the second generation, but it can no longer mate. These particular barriers then keep species from mating. If you look at my picture over here, microevolution would be this first case over here with the arrow. This is where your population's gene pool has changed over time. So you can see the form of the bird has changed, but we have not created any new species. So that is microevolution. Macroevolution is where we start with this one species, but over time, reproductive barriers come into play right here and separate one species from another so that you get a new species. Anytime you see this split in these trees, that's where a new species has formed, and that's where we get these speciation events occurring. Now, there are 2 ways in sort of geology and on planet Earth, and there are a few other ones, but 2 ways that I want to talk about how we like sort of form new species. I want to talk about allopatry speciation and sympatric speciation, these 2 ways of like sort of developing or having these new species evolve over time on planet Earth's surface. So let's talk about allopatry first. In allopatry speciation, you need a geographic barrier. I'm showing the Grand Canyon in my picture here, but it can be other geographic barriers like a flooding river that then when the flood waters recede, you get sort of a lake on sort of the outside of the river so that you're separating a population by some kind of physical geographic barrier. So a canyon, river flooding, it can be mountain ranges coming up and then separating species. These kinds of events will then take one population that used to be all together and splinter it into 2 groups with some kind of physical barrier between them. Over time, if those species are left alone, then when we bring them back together, this can be thousands and thousands of years later, then they no longer recognize each other as mates. Enough genetic sort of differences and mutations have occurred that separate those particular populations. Reproductive barriers are then in place and they no longer recognize each other as mates. So allopatric speciation requires this geographic barrier to separate our populations. Sympatry is a little different. This is shown over here in this picture of this tree species. Remember, allopatry is like this mountain range that splits or splinters, a geographic barrier that splinters a population into 2 groups and they become 2 different species. But in this other picture, we have this tree species that is without a geographic barrier splintering into 2 populations. And how this can work is sometimes an example of this might be like genetics. If this tree species originally has 30 chromosomes, and plants do something where they sometimes create polyploids, which is where they create new sort of chromosome numbers based on inheriting a full extra set of chromosomes. And so if these tree species started with 30 chromosomes, but some of them became polyploids with 60 chromosomes, where they inherited a full set of chromosomes, an extra set, then you could splinter this population and you could have suddenly trees with 60 chromosomes, some with 30 chromosomes, and that chromosome number could keep them from mating, could keep them separated over time so that they no longer can like sort of mate back with one another. This is an example of how sympatry works, where one population splinters into 2 groups without a geographic barrier. So in my example, we're using like sort of chromosome numbers to kind of splinter our population. Now, if we look at geologic timescales like this one shown in our picture, we can see how different species have evolved on the surface of the Earth's surface. We can see extinction events that have taken away certain species, and this gives us really a story from the beginning of sort of Earth origin all the way to present of how different species have changed over time. I don't want you to know all of these particular pieces of this geologic timescale, but I do want you to know a few different pieces, and they're in red at the top here. I do want you to know that the origin of the Earth is, we think Earth is about 4.54 billion years old, and so I want you to know that particular date. Some of these diagrams will show 4.6, but my geologist friends prefer the 4.54 billion years ago sort of date. I also want you to know when the first prokaryotic cells or life is fossilized in our fossil record, and that is listed at about 3.5 billion years ago. And then the first cells that are like your own cells with true nuclei, they're called eukaryotic cells, those come to be about 2 billion years ago. You can read through the rest of this. We have multicellular life, animals, plants, and humans on this picture. And I like this clock because it shows kind of when those organisms have occurred. Humans are at this last kind of second on that particular clock. But this helps us tell a story of how different populations, how species have evolved on Earth's surface, gone extinct perhaps, or changed over time to the present day organisms that we see. Last thing I want to say about speciation is how scientists actually visualize and classify different species into groups. The old school, starting in the 1700s system, was a naming system, and this was Carlo Linnaeus's binomial nomenclature naming system. And Linnaeus's naming system works from very general categories, like our 3 domains of life. We have 3 domains: bacteria, archaeans, and eukarya. And so there's 3 big groups of living things, so very generalistic. Those are called domains, down through a number of classification categories until we get to species, right? Things that can interbreed in nature and produce fertile offspring. So if we go down that, it's like domains are the most general, and then kingdoms come next. Then phyla are a little bit more classified. And if you remember back in high school, you might have learned a rhyme like King Philip came over for good steak. So you go kingdom, phylum, class, order, family, genus, and species. And that genus species is a 2-part name like Homo sapiens that indicate a group of organisms that can interbreed with one another in nature and produce fertile offspring. So this old school naming system grouped organisms into these classification categories from most general to most sort of specific. The new school method is called systematics, and we develop these evolutionary trees called phylogenies. I'm showing one over here. Phylogenies have roots, so that is like sort of the ancestral organism of all the different organisms that we wish to compare. We have an outgroup, or the least related, the first organisms to split off, and then we have over here on the tips all the different organisms that we wish to compare. And these particular evolutionary trees show time sort of going forward, and they show how these groups of organisms are related. And at these nodes, we get our most recent common ancestors. So we can see that ostrich and hawks share a more recent common ancestor than ostrich and crocodiles, which is back in time at number 5. Crocodiles then share an ancestor with lizards and snakes back in time at 4. And so we can talk about who's most closely related, who shares a more recent common ancestor over the others in a particular group. That's all I want to say about speciation and macroevolution. I will see you next time.